Hello everyone, welcome. I'm Jack, the technical support from TransGen. Today, I will walk you through our nucleic acid extraction portfolio. This training is designed to help understand what products we offer, which kit is best for which sample type, and how to position each product for your customer. Let's get started. This slide shows four modules we will cover today: DNA extraction, RNA extraction, plasmid extraction, and automatic extraction. First, I will show you our three extraction technologies. First is the precipitation method. Using this method, it always has lower price, but it requires high requirements on operating skills, which means if the operator is highly skilled, this kit is good. But if the operator is a freshman, maybe this kit is not so friendly for them. And the second method is a column-based method. This method using a silica membrane to bind the nucleic acid. Compared to the precipitation method, it always has simple operation and is much easier and achieve high purity. And the third method is the magnetic beads-based method. This method always achieve high yield and quality, and a big advantage for this kit is that it can be compatible with the extractors. So if the customer has a lot of samples to be extracted, this method will be a good choice because it is high throughput. This is an overview of our nucleic acid DNA extraction products. We categorized the kits according to the sample types, as you can see from plant, blood, bacteria, marine animal, cell tissue, cfDNA, yeast, FFPE, soil and stew, you can see, and also microbiome, all kinds of samples. You can target the kits according to your customer's needs easily. Here we recommend three of our top-selling kits. Each of them contributes more than 1 million RMB to our annual revenue. Genomic DNA Kit EE101, Universal Plant Genomic DNA Kit EE112, and Stew and Soil Genomic DNA Kit EC801. First, let's see the genomic DNA kit. It has a wide range of applications, which means it can extract all kinds of materials like animal tissues, animal cells, E. coli, and yeast. And it uses the enzymatic digestion method. Using this method, you don't need to use the liquid nitrogen or other physical disruption instrument. So it is much easier to operate. And using the column-based method can achieve high purity of the DNA. Here is the data presentation for this case. You can see we extracted DNA from all kinds of sample types, and you can see the gel image of this result. The DNA band is very beautiful, and there's no protein and RNA contamination here. Now this is a slide I'm very proud of. We did this project cooperating with Chinese government. We extracted DNA not only from animal tissues, but also from challenging samples like urine, saliva, and even skin swabs. Here you can see we extracted nearly 8,000 samples. This is a very successful project with the government. The second product is a Universal Plant Genomic DNA Kit, EE112. This kit can extract the DNA from all kinds of plant samples, including those rich in polysaccharides and polyphenols, and also with starches. And here also the performance data for this kit, you can see we extracted DNA from tobacco leaf, sugarcane leaf, and lychee leaf. You can see the tobacco leaf here represents the common plant samples, and the sugarcane leaf represents the samples rich in polysaccharides, and the lychee leaf here represents plant samples rich in both polysaccharides and polyphenols. You can see for all of these three samples, the concentration compared to the competitors is always much higher, and the gel image you can see, the result is very good, single beautiful band, and no protein and RNA contamination. In here, this slide shows the verified sample types. You can see all kinds of plant samples, and here the samples marked with red is that rich in polysaccharides and polyphenols. Both of them achieve very good results. You can see the yield is very high. And the next kit is the slow and soil genomic DNA kit. It can extract DNA from slow and soil. And using this kit, you don't need to use the cumbersome steps such as heating or ice bathing. And here we also include a component here, humic acid removal reagents. These components can efficiently remove inhibitors to help you achieve high-quality DNA. Let's see, here is a verified sample types. For this kit, we validated its performance on soil samples from all over China. In fact, the longest distance between the collection sites is more than 3,000 kilometers. The soil temps varied enormously, but as you can see from the data, EC8001 delivered a high concentration and good quality DNA in every case. And also, we tested the stool samples from different animals. You can see for tiger, pig, wolf, cow, fox, human. You can see from different animals, the stool samples, the DNA extracted is very good. Here is the data performance for soil samples. You can see from farmland or grassland, the concentration is much higher, and we use the DNA extracted with this kit to do the downstream PCR. You can see the PCR result is also very good, means there is no inhibition in the DNA we extracted. And the same data for stool sample, you can see we won't go much deeper into this stage. Now let's move to RNA extraction. We have a very strong portfolio here from plant, blood, cell tissue, and fungi, bacteria, also a solution for mRNA, and also from virus. We will dive deep into these 5 kits here: TransGen Plus RNA kit, TransGen Simple kit, is pure RNA kit, fast cell RNA kit, and universal plant total RNA kit. First, let's see the first RNA kit, ER501. This is our top-selling kit. It has the strongest lysis capability. It is our broadest coverage kit. It can extract RNA from almost any sample type, but the extraction speed is not so fast. You can see here from different sample types like animals, plants, and microorganisms. As I said before, for the ER501, it can extract RNA from all kinds of sample types, but the extraction speed is not so fast. Here we still have a faster extraction kit according to some sample types. Like for this one, ER511, it can just complete it in as little as 8.5 minutes. Also, you don't need to do the centrifugation-based phase separation, so the operation is much easier, and it is very safety, free from toxic reagents such as chloroform. Here we compared these 2 cases. You can see the whole workflow is much easier and no phase separation, easy to pipette, and you don't need to use the toxic reagents. And this slide shows the verified samples. Except the animal tissue cell, it can also operate the marine tissues like clam, oyster, and shrimp. This is the data presentation here. You can see compared to the competitors, our product is better, especially here to extract RNA from rice leaf. And another product is this one, ER101. This kit uses a column-based method and can extract RNA from animal cells, tissues, and E. coli, and also can complete the RNA extraction in half an hour. And here in this kit, we have a DNase-1 in this kit to help you avoid from the gDNA contamination. And here is the data performance from different samples. You can see we compared to the competitors, no matter for the concentration and the quality, both of them are very good and better than the competitors. And the next one is the fast cell RNA kit, ER111. It can extract RNA from the animal cells, and it is the fastest one. It can be completed in only 7 minutes, and it is also very safety. You don't need to use the toxic reagents. Here you can see from this figure, you can see how fast it is compared to the competitors, only 7 minutes. And the quality is also very good. You can see the result here, the yield. The next one is the universal plant total RNA kit, 302. This kit can extract RNA from all kinds of plant samples, especially those rich in polysaccharides, polyphenols, or starches, and the whole workflow can be completed in less than half an hour. And here you can see the performance data here. We extracted the RNA from tea tree leaves, corn root, and soybean seeds, and the concentration is much higher than the competitors. And here you can see the gel image here. The competitor may fail on this experiment. Here you can see we've verified different plant sample types here. Here I have two specific success stories for this kit. First, customer in both Guangxi, China and Brazil use this kit extensively to extract RNA from sugarcane. The feedback is constantly very good, and the RNA is pure and works well for the downstream applications. And second is that we took this product to Malaysia and ran a hands-on workshop. We had 30 technicians using the kit simultaneously, and all of them got good results. This proves how easy and robust the kit is. And the next part is the plasmid DNA isolation kit. Here we have a simple diagram to help you choose which extraction kits you can use. Like for the sample input lower than 20 milliliter, we have EM101 and EM111 mini pipe. And for sample input less than 60 milliliters, we have EM112, the mini pro. This is our new launched products. And for samples between 60 milliliter and 500 milliliter, we have 4 kits here: EM122, EM123, maxi pipe, and EM152, EM153, 1-tube maxi pipe. So what is the difference between this kit? You can see here for these 3 plates, we have 2 for each of them. For the EM111, it has lower endotoxin than this one. So this is more used for transfection, and also the same as this one, has lower endotoxin level. And EM153 also has very low endotoxin level for the usage of transfection. Here shows the detailed data for this kit. You can see compared to the competitors, it saves 50% time than the competitors. So why it is so fast? Because if you use a lot of samples, you may use a large amount of lysase buffer or neutralization buffer. But in our kit, you can just incorporate these reagents into 1 tube. So it is a 1-tube version. It saves a lot of time. And this is the data for this kit, EM153. You can see we extract plasmid from different plasmid culture. No matter for the concentration or the total yield and in the endotoxin level, all of them are very good, with high concentration at low endotoxin levels. And this kit we compare to the Cipquim kit. You can see how low endotoxin it is. So it is very good and suitable for the transfection experiments. Here we compared the plasmid maxi pipe products. You can see for the sample input between 100 to 200 milliliter, we have 2 kits here. And for sample input between 200 to 500, we have EM152 and EM153 here. This kit is endotoxin free and used for transfection. You can see the detailed information here. And the next part, we will introduce you the automatic extractors. You can see these 3 pictures is our automatic extractors, TS16, TS32, and TS96. For this one, it can operate 16 samples simultaneously. This one, 32 samples simultaneously. This one, 96 samples simultaneously. All you need to do is to touch the screen here and put your plate into the instrument and click start. So the whole workflow is very easy, efficient, and also very intelligent. And here we listed the kits compatible with the extractors. You can see for DNA, RNA, and co-extraction of DNA and RNA from different sample types for cell tissue, soil stool, and plants, almost tissue FFPE, plasmid, plant, and also from clinical samples like blood or serum plasma and liquefied sputum, a swab, all kinds of samples here. You can find the according kits. Here you will find a comprehensive list of references and publications in these slides, showing the strong scientific backing of our projects. We put all the references here for today's project. Thank you for your attention. I hope this gives you a clear picture of our comprehensive and reliable nucleic acid extraction portfolio. Thank you.